Libo.
You're hearing the sound of a Parisian pedestrian crossing signal. It's saying the light is green, you may cross. It's designed to help the blind cross the street safely. And a few years ago, something happened at one of these crosswalks, an event that was arguably a watershed moment for science and for all of us. This episode is about optogenetics and how implanting genes found in other organisms from the ocean into humans allowed vision-impaired people to basically borrow their eyesight.
Alors, j'ai appris ça, j'avais 26 ans. Et mes parents m'ont envoyé voir un ophtalmo, alors naturellement,
I got the diagnosis at 26. My parents sent me to an ophthalmologist. He corrected a slight myopia, and that was the end of it. A few years later, I went to Paris again for an eye exam and learned that it wasn't just nearsightedness, but most likely retinopathy or retinitis pigmentosa.
The man speaking is a former sailor who lives in Brittany, a northwestern region of France, and he is 58 years old and has been completely blind for the past 16 years. We don't know the name of this volunteer, but we do know exactly what happened to his eyes. Retinitis pigmentosa is a diagnosis that has left about 1.5 million people worldwide living with very poor vision or no vision at all. Here's how it works. The retina of our eyes is similar to the sensor of a digital camera, so specialized cells, they are called rods and cones. These cells are arranged in rows in the retina like dots on a camera sensor, and these cells are sensitive to light. So in response to illumination, they trigger an electrical impulse. Let's say any image has darker and lighter areas. Imagine the letter O drawn on a black background with white paint. The light emanating from the bright areas of the image illuminates the dots on the retina, and the pattern of illumination forms that shape of the letter O on the retina, while the remaining dots stay dark. And beneath the layer of light-sensitive cells lies the next layer. It's called layer of ganglion cells. And these cells read the electrical impulses and they relay those impulses to the brain via the optic nerve. And then the brain interprets these signals. Okay, so this comes from the retina, so probably it isn't something sour or cold that I'm sensing. And then I, the brain, need to assemble those impulses into an image, the letter O. But in retinitis pigmentosa, the rods and cones die. The retina no longer responds to light, and blindness sets in. And the reason I'm talking so long about that technical stuff is this. Despite all the massive cell death in case of this French sailor, that very last ganglion layer remains intact, with live cells. And this is the layer that scientists would later on in this episode attempt to make sensitive to light again. But for now, let's listen to the patient.
I remember at one point I found myself standing on the tracks in the metro. After that, the doctor said, "Listen, it's time to wrap this up. I'm going to give you medical leave. Otherwise, you're going to seriously hurt yourself." And I've been on leave ever since.
So this is a heritable disease. Individuals develop it if they inherit 2 defective genes from their parents. Symptom onset happens at a young age. The dots, the light-sensitive cells in the eye, start to break down. In the case of patients with the most severe form of this disease, like our French sailor, this layer gets fully destroyed and distinguishing light from darkness becomes impossible. It's like a digital camera which has a burned-out sensor, but the connectors are still intact. Is it possible to use what is left to start making images again and relay them to the computer, that is, the brain. It sounds impossible. What is broken is broken. But there was one man who came up with a crazy idea.
I'm Jose-Alain. I'm an ophthalmologist. I'm the chair of ophthalmology at the University of Pittsburgh Medical Center and also a professor at Sorbonne University. My area is retina and especially retinal degeneration. So it started around 15 years ago. I heard about the discovery of these proteins in algae that was made by very basic scientists in Germany in the late 90s and early 2000s. And at that time, the feeling was that this was a great tool, first for marine biologists, and second for neuroscientists that want to use that to manipulate neurons and circuits in the brain and everywhere.
The protein that Sahel mentions are light sensitive. Single-celled algae, a type of plankton, float near the ocean's surface, beating their tails to propel themselves in the direction of the sun. And if you ever took a dunk in the ocean, you have swum with these plankton. On the surface of their cells sits a special sensor that allows them to swim in the right direction. It works a little like a photosensor on an automatic door, a little peephole that detects light. But only in the blue part of the light spectrum. That's important, remember it for later. When it catches a beam of light, it opens a channel in the cell membrane, and that triggers the cell to speed up the beating of its tail. So the algae swim toward the light, which is food for them because they photosynthesize. And here comes the really cool part. If we leave the vision aside for a moment, there is a really magical technique based on these photosensors that allows us now to study what happens in the brain. Because using the technique gives you control over it, kind of like a TV remote control. Here's how it works. There's this photoreceptor in the algae. So there is also a gene that encodes it so that the algae cell can produce that photoreceptor. Scientists have isolated this gene and learned to implant it into various cells in different organisms, like fruit flies or mice or rats. And therefore, these photoreceptors can be implanted and grown on the surface of mouse brain cells, neurons. Then, researchers shine a blue light through a hole in the mouse's skull. This blue light activates the photoreceptors, the channels open, electrical activation begins, and the targeted neurons fire. Using this method, scientists have been able to control mouse behavior, implant mice with false memories, treat them for cocaine addiction, or make fruit flies perceive that they've eaten something sweet. Like I said, it's magical. That's what optogenetics is, using genes to make various cells photosensitive. But all of this is fundamental science. That is, science meant to make discoveries and increase understanding, not to solve patients' problems.
So, and around 2006, at the big vision conference that takes place every year, I met with a friend and colleague of mine who is much younger than me, Botan Roska, who was already using these proteins to study circuits, like everyone in neuroscience. And well, he was giving a presentation and he jotted the idea that maybe we could try that for vision restoration, which is something I thought also that would be a great idea.
So, medical doctor Jose-Alain, an ophthalmologist, was at this conference listening to biologists describing lab experiments on fly and mouse neurons. This, by the way, is remarkable in itself. In many places, it's rare for physicians to be so deeply engaged in fundamental research. But this physician was very curious to hear about experiments with mice, and he immediately thought of his patients. And at first glance, his idea sounds pretty straightforward. What if, instead of making mouse neurons sensitive to light, we could make the retinal cells of a blind person sensitive to light? Why not grow those tiny eyes from algae on the surface of human retinal cells and make them light sensitive again. Problem solved.
And everyone in the room was laughing at that. People were saying, this is so ridiculous, it will never work.
The skeptics had some valid concerns. Photoreceptors from algae respond only to very intense light, and only blue light, the part of the light spectrum algae actually need for photosynthesis. But light is a wave, and waves have wavelengths. You probably know that waves with very short wavelengths are ultraviolet, and ultraviolet light carries so much energy that it can burn your skin, so you wear sunscreen. Blue light is less aggressive, but its wavelength is also relatively short in the quantity that's needed to trigger photoreceptors from algae. Blue light is simply dangerous. Even if there was a mechanism to deliver that amount of blue light into the eye, you'd end up burning it to cinders, like if you had stared at the sun in the desert on a perfectly clear day. It's that bad. Longer wavelengths correspond to red light, which is lower energy. So, on their own, genes from algae cannot restore a blind person's sight. But there was one person in the room who thought this might be worth literally looking at. Remember that friend Dr. Sahel mentioned, doctor and scientist named Botan Droska?
So after the meeting, after the presentation, we sat together. We are very good friends, and I've known him since he was a graduate student almost, so really very bright. And so we sat together, and I told him, I don't think it's stupid at all. I think it's really something we should try. And so we decided that the best way to know if something is not going to work is to try to make it happen, and then we'll know whether it works or it doesn't work.
So the main challenge and the main task here, if you actually want to apply all this magical science, is this. You need to build a system that, instead of blue light, can work with much weaker light and with longer wavelength. Fortunately, while Sahel was doing his work, fruit fly scientists, actually a whole swarm of fruit fly scientists, were moving in the same direction. They were also in the market for photoreceptors with a different range of properties to design even more intricate experiments with their fruit flies. And this set off explosive growth in the field. Biologists examined 120 species of algae from the world's oceans in search of photoreceptors with differing characteristics. And they identified several dozen that were worth looking into further. And the work didn't stop there. Scientists began modifying these photoreceptors, creating mutant variants by playing with the letters in their genes, eventually assembling an entire collection. Now they had photoreceptors sensitive to different wavelengths of light, so to different colors. For example, scientists developed an intriguing photoreceptor called E2 crimson, which could have practical applications for physicians. Because this photoreceptor responds not to blue light, but to yellowish-brown light, the color of amber. And this light has a longer wavelength, making its radiation gentler and less harmful to the retina. So, Sahel took that crimson photoreceptor and started to use it in experiments with human patients.
We did many studies, actually studies with mouse retina, rat retina, primate retina, ex vivo human retina. So taking the retina post-mortem from donor eyes and looking at the efficiency.
Once they were confident that they had a working system in hand, the scientists began selecting volunteers. They looked for people who had gone blind in adulthood, and only those who were completely blind. Because if any vision remained, there was no guarantee that the new system would produce a noticeable improvement in their quality of life. In 2019, the Paris Vision Institute, then headed by Sahel, recruited the first 8 volunteers. Each volunteer received an injection into the center of one eye using a virus, similar to the type used in COVID vaccines. The virus delivered genes from algae into the surviving base layer of the retina. This moment is really remarkable. People growing something like that in their eye. 4 to 7 months later, doctors were able to look through the pupil and see if the necessary photoreceptors had developed. How could the doctors tell? Well, amazingly, the virus didn't deliver just the gene from algae. This gene was actually a chimera. That means it combines the gene from algae with another one from a certain bright red coral. As a result, chimeric photoreceptors were formed. Each photoreceptor had an extra little tail that glowed with a beautiful tomato red color under ultraviolet light, earning it the name D-tomato. So, by shining an ultraviolet flashlight into the eye, doctors could see they played God well enough. This tomato red glow on the retina, tiny red dots that confirmed that photoreceptors had grown. How long would they last in the retina? Well, judging by experiments in dogs, the photoreceptors should live a minimum of 10 to 20 years. And as Sahel told me, it's no problem to inject some more of the virus later on to update the genetic instructions for the retina so that it grows more photoreceptors. A huge further challenge, however, lies elsewhere. Yes, you are the blind person, you've grown these photoreceptors, doctors are happy, this experiment is already half successful, but unfortunately, despite all that, you still aren't able to see anything.
Yeah, so the patient, so this specific patient, hopefully we'll have more to report soon, but this specific patient has been affected with the disease for 40 years, since he was a teenager. He became blind when he was 40-something.
40-something?
42 or 43, yeah. Mm-hmm. And now he's 58. Mm-hmm. So when he got the treatment, we didn't promise anything to him. We told him this is totally experimental. We just check the safety. And almost the first year, close to 1 year, he was a bit disappointed because nothing was happening. And now he tells us that he was starting to see some light, but he couldn't make sense out of it.
Why was this happening?
In a normal life, the human eye is adapting to many levels of light. We adapt to almost darkness, and we can also see at very bright environments. So it's like 12 log units of lighting. And so this is what the human eye is able to do. But to do that, the human eye is using hundreds of proteins, hundreds of adaptive systems that are working from that, which is just unbelievable. And we don't even know all of them. But the protein we were planning to use is only one protein. So it can't replace thousands of protein or hundreds of protein. And it's working only in a very specific range of light. So the idea is that if we use a camera that can work at many levels of light, then we could adapt to that.
So again, after being injected with the gene, people were not able to see anything. There were a lot of problems. The levels of light needed to be much more moderate than in real life. It all had to be amber shaded. And there was a whole other problem with the fact that things are not sitting still. They're moving. So, next up, if they were to have any hope of being able to see, it would be with the help of a very special gadget. Imagine a person wearing special glasses, a little like VR goggles. There's a camera lens sitting on the person's nose bridge. In front of the eye that was injected with the virus, with the photoreceptor gene, is a special kind of screen, a tiny powerful projector. This projector is live streaming a very special kind of image from the camera into the eye using its built-in processor. This image, firstly, is entirely amber colored because, remember, photoreceptors can only detect amber light. And second, the brightness is balanced. The camera can record video in both very bright sunlight and dimly lit rooms, but the projector displays a moderate version that the reprogrammed retina can interpret. But that's not all. The transmitted image conveys motion in a very unusual way. The projector doesn't just show standard video like on a TV with 24 frames shown per second. Instead, it sends a special type of video signal that more closely mimics the way a real retina processes information. We've talked before about the eye as a camera, a metaphor that's been in use for 150 years. Now let's talk about the ways the eye is not like a camera. When the retina of the real eye captures the world using its dots, it actually works very efficiently because it only picks up changes in light and ignores any visual stimulus that's not changing. This means the camera mounted on the glasses also doesn't need to capture everything, just the changes meaningful to our brain. So by the time Sahel's group began working with volunteers, this kind of bionic, bio-inspired camera already existed. Let's listen to him.
The other idea is that we need to speak the language that the retina is speaking. The retina is responding to nature by signals. So it's like a music or it's like a signal. And the main thing that is happening in the retina, like in the brain, is that in contrast to photography or movies that we are filming, we are only interested in changes. When you take a photography or you take a movie, you have hundreds of images that take pictures of the wall, of many things that don't change at all. But you take all these pictures and they are useless because nothing is happening. And the visual system is very efficient because it only is interested in anything that is changing. If the image is totally still, there is no image at all. The reason we see still images is that because our eyes are moving a little bit to create some movement. If not, we would see nothing for still images. So the idea was to use cameras that would be working the same way. And this is called event-based camera. So each pixel of the camera is responding to a change. It's called an event. So it's called event-based.
Let that sink in. Isn't it crazy that the eye needs to make small movements all the time in order to see still objects? Otherwise, they would go completely unnoticed for the brain. If you type the words event-based camera into YouTube, you'll see what this looks like. For example, you can see a person standing in a room. The entire room appears entirely gray because nothing is changing, so nothing is being registered by the camera's sensors. But when that person moves, even slightly, their silhouette and facial contours appear against the gray background in sharp, high-contrast black lines.
Just to give you a side comparison to that, people are using cameras to monitor intrusion into a building, for example, like detection cameras. They keep taking pictures all the time and nothing is happening. Nothing is happening. Nothing. The walls are still. So if you use the type of cameras I'm mentioning, there is no image unless something is happening. So instead of taking tons of images that are being stored on servers and that feeding all this internet and everything, you only detect a change when a change is occurring. So this is how the retina is working.
Blind mice whose retinas were modified with optogenetics and who wore the same but mouse-sized event-based cameras were successfully trained to see this way. But mice can't tell us what they're feeling. Sahel's group still had to find a human willing to learn this new visual language after many years of blindness. And this was very, very difficult.
And this is where you get into the third part of it, which is the training, because the type of image that you are producing is not natural images. These are very strange images, and the patients have to learn how to make sense out of that.
So, again, first of all, they needed to confirm that the necessary genes were working. They could see that wonderful D-tomato glow in people's eyes, which means their eyes are growing these photoreceptors. And then the French doctors prepared to move on to experiments with a special wearable device. Remember? The one which looks a little like VR goggles and feeds the eye with a special event-based, amber-tinted video. And then the pandemic hit. Amid 3 waves of lockdowns, only one patient was able to complete all the stages of training, that sailor we started our story with. Still, enough patients stuck around long enough that the researchers were able to tell us how these totally blind people reacted to the projector in their glasses being turned on for the first time. Many felt very anxious, unable to find words to describe what they were feeling. Some began to sob. Imagine the brain hasn't created any images in years, and now the retina is sending the brain this strange signal. It's clear that since it comes from the retina, it's not telling the brain that something is sour or cold. It must be a visual signal, But of what exactly?
First, they have to learn how to align the beam into the eye. And second, they have to understand what they see. And the patient we are describing in this paper, he could see changes, exactly what I described. He could see the edges of the table, could see the edges of the object, but he had to make sense out of it. And so there is a training process that takes place. And so this is the third part of the technology, which is really the adaptation of the system to humans and the humans to the system. And because the system is totally tunable, because it's outside the eye, you can reprogram to make it meaningful for the patient. So the patient had to be trained. This is why it took some time to get there.
The camera responds to changes. So the best way to use it is to seek out the outlines of objects while moving your head slightly from side to side. Just like in a normal person, the eye is moving constantly. The movement makes the outlines of objects shift a little and makes that area become a site of change. So you begin receiving visual information, like, for example, information about white stripes on a pedestrian crosswalk, which is exactly what happened with that French sailor when he first tried the camera and projector glasses.
Le premier sentiment quand j'ai effectivement...
I remember that in one of the experiments, I was outside searching for a crosswalk. That's when I realized I could actually locate the crosswalk when I had the device on my eyes, and that I couldn't find it again right after when we repeated the experiment without the glasses. It awakened something in my eyes. I told myself, no, I can't see, but my brain can. It produces very peculiar images. When I say that I see things flickering, I mean that I feel a vibrating light.
And well, obviously, when you do all of that, you hope it will work. But a lot of things we do in science never work. We worked 5 years, sometimes 10 years. And at the end of it, you publish a couple of papers, but it doesn't work. So until we knew that the patient was telling us he sees something, we were just hoping for it. And so the day it happened, it was a major event for us, obviously.
Have you been there in Paris when it happened?
Actually, no, I was in the US. I got a phone call and someone told me, he sees, the patient sees. And I thought it was another trial. I didn't think it was this trial because I have many trials ongoing, many technologies. And when I realized this was this patient, well, it's like all of a sudden, all the history makes sense. Like all the dots are becoming aligned at 1 minute. But then I thought, okay, and so they wanted to do a press release. I said, no, we don't do a press release. We check everything again and again. So we spent a year checking everything, recording from the brain in the patient, doing different types of testing, and importantly, publishing in the best possible journal to make it a thing that nobody could argue that it's a fake story. Fake news.
And so, this French patient, whose name we do not know because the study kept his identity anonymous, this patient became the first person to learn to see again with the help of optogenetics. His brain began to learn to distinguish object contours. There is a video I shared on the Libo YouTube channel in which the patient takes a bottle of hand sanitizer out of his pocket, an item he became very familiar with by touch during the pandemic. And he sets that sanitizer on the table.
Là, j'ai un deuxième qui est pas le... Non, c'est pas l'extérieur, j'en ai un deuxième.
And now sanitizer is standing on the table, a meter away from the camera, perched on his nose.
Là, j'ai un truc que je vois, je ressens, je sais pas vous dire si... Non, à mon avis, il est sur la première ligne.
And for the first time ever, a human being describes to the scientists in the room what's it like to have photoreceptors from algae in the retina, reading events signaled by a projector's light beam. And now you have to turn these signals into information about the right and left edges of the familiar little bottle of sanitizer.
For example, he told me that the goggles we made were misaligned by 15 degrees because this patient was a sailor. So he knows exactly how to measure the angles and everything. So he said you have to realign your beam by 15 degrees to get where you want it to be. Quite precise.
A man who hadn't been able to see anything for 15 years was able to look in a mirror and see that his own hair had gone gray. And now, like a true Frenchman, he's hoping that his newly regained sight will help him with his cooking.
Alors j'ai une idée, c'est effectivement quand je fais la cuisine.
I have an idea. This might be helpful when I'm cooking. In one of the experiments, I managed to find the induction cooktop. Why? Because it's black against a white background. I need to keep practicing to understand whether there's progress, whether I'm getting better. Then I need to train so I can adapt this system to myself, personalize it. When I think about the results for me or for someone like me who has mourned the loss of their sight, it's a miracle that I can now say to myself, well, I can see again. There's so much hope in that.
And there's so much hope in that for us as well. A small step for one blind patient, a giant leap for humanity. One of Earth's inhabitants has, for the first time in the history of our civilization, had a chimeric algae coral gene implanted in his retina, and with the help of a bionic video camera, learned to do something he could not do before. Not only will we conquer blindness, optogenetics promises a myriad interventions into the workings of the brain and other organs. We will continue sourcing genes and super abilities from different organisms and using them to treat various diseases. This was The Naked Mole Rat, a Libo Studio podcast. To help more people discover us, follow us wherever you get your podcasts, leave comments, and give us ratings. It helps more people hear about us. The Naked Mole Rat is produced by Andrey Borzenko, Ilya Marritz, Danil Astapov, Anastasia Medvedeva, and Lika Kremer. Our sound editor is Ildar Fattakhov, and I'm Ilya Kolmanovsky. See you in 2 weeks.